Okay, so we're going to move on today from the adrenal gland to talk about another very important gland and a series of diseases following this that are probably the most costly and the most prevalent, or certainly linked to one of the most prevalent problems globally, and that is having to do with pancreas, insulin levels, sugar levels, and more importantly, diabetes. Diabetes, we'll talk about how the pancreas works. We'll talk about the hormones that the pancreas is responsible for producing. We'll talk about how these are controlled by other parts of the body. We'll then move into type 1 diabetes, what type 1 diabetes is, how it's different than type 2 diabetes, and then how the two diabetes are controlled. The pancreas is often underappreciated for how important it is, and these two disorders, well, type 2 in particular, is certainly one of the most expensive and leads to massive comorbidity throughout the world. And it's a rising problem as we all eat too much fast food. What's the fast food, the chicken chain in China that's so good? I forget the name of it. They have fried chicken and chicken sandwiches. Oh, so good! I love that place. Too much, but eat too much of that, and obviously you'll start to have problems with your weight and then your pancreas and your sugar levels. So let me start the presentation. Can you see the slides? Okay, great. Yeah. So some of the things we're going to cover. I want you to be able to list the hormones. So if I ask you a question, what hormones are produced by the pancreas, you'd be able to tell me what those are and be able to tell me the cell types that are responsible for secreting them, because the pancreas, very much like the adrenal gland and say the hypothalamus, is made up of very specific cell types that are responsible for very specific hormones. Okay. And also remember the thyroid also had that issue. So that's an important theme I want you to be able to replicate when it comes to the exam. I want you to be able to describe the biosynthesis of the peptide hormones, particularly when it comes to insulin. I want you to be able to describe the process of insulin secretion, its mechanism of action, and its biological functions. And particularly, those of you who paid attention to my previous exams, I want you to be able to talk a little bit about the receptors. Okay, so for insulin, it's a tyrosine kinase receptor. So you should be able to draw or describe to me how insulin is produced, how its biosynthesis occurs, how it binds to its receptor, and then how this receptor signals and leads to changes, physiologic changes. I want you to be able to describe the synthesis, secretion, and mechanisms of actions and functions also of another major hormone that we'll talk about, and that is glucagon. I want you to be able to talk about the gross anatomy of the endocrine pancreas, what it looks like. I want you to talk about synthesis of these peptides, because we'll cover a bit about that today. Then we're going to talk about hormone diversity, because similar to some of the other hormones we talk about, sometimes you get the sharing of the gene, or genes are very similar and diverge. Then we're going to cover the mechanisms of glucose-induced insulin secretion and the regulation of insulin secretion. And then finally, as we start to get closer to understanding the diseases, we'll talk first about what are the mechanisms of action and biological function of insulin.
Sorry.
Yes. Yeah. Apologies. I'll go back. So just to cover what I want you to be able to know, because this is an important slide. I want you to be able to list the hormones secreted by the endocrine pancreas, and specifically the cell types that secrete them, because as I mentioned, similar to the hypothalamus or the thyroid or the adrenal gland, you have very specific cell types controlling very specific hormone production and secretion. So you should know what those slides are, those cells are. You should be able to describe the biosynthesis of peptide hormones, particularly in insulin and particularly in glucagon. And in addition, as I mentioned, please know how the receptors work. So the glucagon, so the insulin is a tyrosine kinase receptor, whereas the glucagon is a G protein coupled receptor. Okay. All right. So this is how we'll cover the next few series of lectures. There's quite a bit of content, so I'll try to keep it in small bites and not make you eat too much in one day, if you know what I mean. So we'll start first with the gross anatomy of the endocrine pancreas, and then we'll move on from there. So this is what the pancreas looks like. And you can see it links very closely with the small intestine. And it has, like some of the other organs we've talked about, it has major blood vessels and arteries going through it. It also has this very important pancreatic duct running up the middle of it, which is crucial for the endocrine portion of the function of the organ. The pancreas is responsible for secreting digestive enzymes directly into the GI tract, into the small intestine. And that is its exocrine function, okay? And we won't talk too much about its exocrine function, but we will focus on the release of insulin into these blood vessels, which are efferent throughout the organ. Insulin is important for regulating the blood glucose levels, concentrations, and glucose utilization throughout the body. So I want you to be able to remember what the difference between endocrine and exocrine glands are. That's one thing we covered in some of the earlier lectures. Visually, you can see that the two functions of the organ, exocrine and endocrine, are separated physically. You have this exocrine portions of the pancreas that are linked directly to the small intestine, and then you've got the pancreatic islets, which are driving the endocrine portion. And if you see here, I don't know if you can see my cursor or not, but if you see here, this blood vessel coming in is the crucial part, which allow coming in and going out for the artery. Is crucial for the delivery of the endocrine function or insulin. And you can see this very patterned tile image of the pancreas, and you'll see this in pathology. This will help identify right away when someone has diabetes, because you start to see this tiled structure or formation break down and not be in a uniform manner. So just to locate the pancreas within the overall anatomy, it is located just above the stomach, and which is also linked, as I mentioned, to the small intestine, small intestine, and large intestine surrounding it. It is also just below the liver on the right side torso. It stretches diagonally across the torso and the abdomen. And it's obviously located perfectly to be able to interact and communicate with all of these other organs, which it does. Now, visually, the pancreas is shaped a little bit like a candy cane or a walking cane. So, or a hook. It's got this head region of the hook. It's got this neck region, which is made up of the neck on this side and what's called the ussanate process on this side. And it's got this long body that extends down with a very small tail. So if you remember it as a hook, then you probably won't go wrong. Now, as I mentioned to you, it's got a very unique patterning of cells. And one of the major cells within it, the pancreas, are called islet cells. And these islet cells were discovered by a gentleman named Paul Langerhans in the 1800s. And they have a very unique patterning, as I mentioned to you. So they look, this is what the islet cells look like. You can see they look like little islands within a giant shape or circle. And what's interesting is each islet cell is specific for a different hormone, which we'll talk about now. So, and each one is made up of, the pancreas itself is made up of different percentages of these islet cells. So the first cell type is called alpha islet cells, and this is roughly 15 to 20%, and they are responsible for producing glucagon. The second type of cells are beta islet cells, and they are responsible for 65 to 80% of total islet cells, and they are the main cell type that we'll talk about when we talk about insulin and insulin regulation. They secrete insulin, proinsulin, C-peptide, which we will talk about, and then a hormone called amylin. The delta islet cells, shown here in black, make up about 10% of the total cell type in the pancreas, and they are responsible for secreting a very important hormone, somatostatin, which we I don't know if we covered somatostatin compounds already. I can't remember, but if not, we'll talk a little bit about that in the hypothalamus and pituitary disease treatment. Finally, there are two other pancreatic cell types. One is thought to be called the gamma or F or PP cells, and these are responsible for producing pancreatic polypeptide. And finally, you've got the epsilon islet cells, which are thought, a very small percentage, which are thought to produce the hormone ghrelin. Now, ghrelin is responsible for controlling appetite. It's one of the very important regulators of appetite and communicating with the brain when food is taken in and needs to be processed. So there's a lot of interest in targeting ghrelin for appetite control and suppression from the gut. But it's interesting because ghrelin has to be modulated so that it can cross the blood-brain barrier. So there's a lot of interest in this gut-to-brain communication via the ghrelin system. So visually, what does this tiling look like, and how do all these cells function? So you've got, as I mentioned to you, blood flowing in from the center to the periphery, because of those blood vessels are efferent and perforating the center of the organ. You've got blood coming in, and you see the blood then flows out. Okay, a little bit like a waterfall or more of a stream at the top of a mountain. So the water comes to the top of the mountain and then flows down gradually. And as the water flows down, these cells are adding things to the water, right? And that allows that endocrine function. So again, you can see here and appreciate you've got your beta cells producing the insulin. You've got the delta cells producing the somatostatin. You've got the alpha cells producing the glucagon. And then this would enter the bloodstream, and then gradually the blood would take these throughout different parts of the body. Now, this image, or this cross section I'm showing you, is equivalent to this long body of the pancreas. Okay, so this is where you can find this image. It's all in this long portion. All right. And importantly, this bile duct up here is at the top of the hook. Okay. And this is where the exocrine function then communicates with the GI tract. Now, what does this look like in terms of pathology visually? So this is a classic pathological slide showing you what a pancreatic islet looks like. It's a section of normal pancreas stained with hematoxylin as well as eosin, just a counter stain. And as observed by Paul Langerhans, the islets in the center are identified very easily by their distinct morphology. Okay, so you have this very distinct patterning in circles, and that of the surrounding exocrine tissue. Okay, so you've got this tiling of the exocrine tissue around these nice tight beta islets. Now one of the first ways to look for a functioning pancreas is to stain for the hormones that are being produced. And just as I mentioned to you, that these are very specific, you can stain for insulin and see the same patterning, right, that I showed you in the previous slide. So you can clearly see that insulin is only produced by a very tight, small subset of cells. And then glucagon is not produced by those same cells. It's produced by alternative cells around it. Okay, so it's not produced by those beta cells. I think visually you can see this quite nicely with these nice slides. Now, this is staining specific markers, not for the hormone, but for the actual cell types, just showing you that insulin is indeed made by those beta cells and that glucagon is made by those alpha cells. And this is one of the key experiments that was performed to really nail down and demonstrate that insulin is made by one cell type and glucagon was made by another cell type. And then some of you did your posters on the Nobel laureates responsible for these hormones, and you found that they did transplant experiments to validate and demonstrate the importance of these hormones in regulating sugar levels and function. At first they did these in animals. So we've talked a bit about the gross anatomy of the endocrine pancreas. And we've talked about that long body where we can find these multiple cell types and nice tiled patterning. And let's talk now a little bit about when we've got these specific cell types and these specific hormones, how are these synthesized within these cells, and what is the process for biosynthesis of these hormones? Because understanding that will allow us to appreciate the different drugs that have been used to mimic or block or replace some of these hormones. So just a quick reminder of protein/peptide hormones and stereogenic cell types, okay? Remember when we were talking about peptide hormones versus steroid-based hormones, one of them requires gene transcription, right, and translation. Another one is produced basically from cholesterol and is an enzymatic process. On the left here, and because of that, you have very specific cell types that are optimized for these two types of hormone production. And on the left here, I'm showing you why that is the case for protein and peptide secreting cells. So you can see that in these cells, they've got a lot of rough endoplasmic reticulum. So just to remind you, that you remember you have DNA encoded in the nucleus. You then get transcription to drive messenger RNA, and this messenger RNA is secreted into cytosol. And on these rough endoplasmic reticulums, they're called rough endoplasmic reticulums. You can remember because you've got these ribosomes that are making protein sitting on the endoplasmic reticulum, ready to put them in the sorting machinery of the cell. You can think about the sorting machinery, right, as sort of the China Post, or when you order something from what is it, Alibaba? No, what's the name of it? I forget the name of the big company purchase things in China online a lot. Oh man, too early in the morning. Too long I wasn't in China. Terrible, right? When you buy something online, right? And then it gets packaged and mailed to you in Nanchang, right? Imagine it's down in Shanghai, or and then it gets put into a truck or a train and then sent over to Nanchang. Well, this process is optimized in this cell type for a specific type of package, okay? And the same thing occurs in the cell, where the cell is optimized to produce hormones by making a lot of endoplasmic reticulum. It's got a lot of ribosomes, and it's ready to make a lot of hormone. These hormones then get packaged in the ER and then move through the Golgi. Just to remind you, I know this is all your review from cell biology from earlier years, but it's important to remember and why this is important when it comes to the endocrine process. So then within the Golgi, you get some of the post-translational modifications that we'll talk about. They then get packaged in specific packages. So this might be the train that arrives from Shanghai to Nanchang, and then from there, the final truck that leads to bring it to your house. These secretory granules, this whole process is regulated, including the secretory granules, and you get exocitosis of these cells into, say, the blood that was flowing down the top of the pancreas in the case. So these optimized cells are called protein or peptide secreting cells, and you can quickly identify them because they've got tons of these rough endoplasmic reticulums, and they've got lysosomes and quite a bit of mitochondria because they need all the energy to make all this protein and all this hormone. Now, on the other side, just visually to remind yourself, are the steroidogenic cells. And in this case, these are optimized for producing cholesterol-based hormones. Now, because of the way cholesterol is made, it doesn't require gene transcription, so we don't need near as much rough endoplasmic reticulum. We don't need as much of the machinery to make protein, but we do need a massive place to store cholesterol, okay? So they have these large lipid droplet type things to handle and maintain lipid content, so you can quickly recognize them under the microscope, and you can stain for stains that will visualize the lipid droplets over, say, the rough ER. And then when this cholesterol is stored in there and then handled by and processed by all the various enzymes that you've heard about for making and secreting our steroid-based hormones. So this is just an important reminder of the different optimized cell types for the hormones that we have been talking about and we will continue to talk about. But this, for example, is an excellent exam question. Compare and contrast these two cell types. Now, just highlighting to you here the key differences that I mentioned to you. So one of them, right, I didn't mention was the smooth ER that you see in the steroidogenic cells, which you don't really find in the peptide protein secreting cells. Lipid droplets are the major marker, but certainly the smooth ER would be visually very different than the rough ER, okay? So this is just what a slide of that stereogenic endocrine cell would look like, and you can see very nicely here these lipid droplets, right? And this is the mitochondria, but it's quite striking, right? This lipid droplet within the stereogenic endocrine cell. As I mentioned to you, this contrasts very clearly with the rough ER and the large Golgi that you'll see in the protein secreting cells. And obviously, as I mentioned to you, these optimized delivery systems within these cells for delivering, including one I didn't talk about, which is the microtubules or microfilaments. And these you can think about as I mentioned to you, these trains that come in from Shanghai to Nanchang, they use tracks, right? Well, the same similar, or highways, and the same similar system exists in the cell, where they've got these microtubules and microfilaments that serve as train tracks or highways to help deliver all the things you're looking for from from your delivery that you ordered from. Was it Tybo? What's the name of the purchasing system? Tybo, right? Or the big. I bought some stuff from there. That's why I can't remember. It's driving me crazy. All right. What visually under the microscope does the endocrine cell look like for these peptidergic endocrine cells? And this is what you can see. And what are these little black dots? Well, those are those little vesicles that I mentioned to you, the little trucks and cars and whatnot that are doing the delivery of the peptide hormone and secreting those out into the surrounding blood. Okay, so you can see them being released here, and they get released to very specific sites, which are heavily regulated. And then you can see here some of the lysosomes that are also responsible for helping recycle and turn over those vesicles. Okay, so the cell, a peptide, both cell types are incredibly busy, right? It's like a massive warehouse of organization, but they're organized in very different ways. One is these nice vesicles, and the other has got those massive lipid droplets where they're sending out steroid-based hormones. So just again to remind you that the peptide hormone synthesis is transcription-based, right? So we've got that DNA to immature messenger RNA. We've then got post-transcriptional processing, which is this maturation of the messenger RNA. And these days we understand a lot more about how messenger RNA is matured. We appreciate there's a lot more components that go into regulating and producing messenger RNA that when I was studying many years ago was not appreciated. There's also been a tremendous explosion of understanding how we may go from messenger RNA to peptide, and that's obviously the process of translation. And these days it's appreciated that even at the ribosomal level, you have incredible control of this translation process. For post-translational modifications, you have peptidases that are located throughout the Golgi, some also in the ER, and some also in the vesicles, the endocytic system, for covalently modulating or glycosylating the hormones. And this becomes important for facilitating folding of the hormones. This becomes important for protection of the hormones when they're secreted. Remember that peptides can get degraded in the bloodstream and the serum, so you need to protect them by adding glycosylation groups or proper folding by, some of these have cysteines, and you need to form disulfide bonds. They're very important. Isomerase is located in the ER and throughout the endocytic system for helping maintain these folds. You then have other proteins that are responsible for controlling the pH. pH changes throughout the endocytic system. So you have a lot of very important modifications that get added on as your package comes from Shanghai. You get, it gets put in a box, it gets taped together, it gets your name put on it, it gets labeled, all of the same things that happen to hormones. So visually, this is what peptide hormone looks like in some of the steps. I've talked about most of them, but just important that you be able to replicate the slide if I were to ask you that question. So You have initial transcription factors that help trigger formation and binding of the RNA polymerase. Polymerase binds to the promoter region of the particular DNA. It drives transcription. Importantly, and we're going to talk about this shortly, is remember when you have transcription, you have these introns and exons. And depending on the processing of this RNA, you will get different messenger RNAs, which can lead to different hormones, even though we started from the same gene. So we'll talk about this shortly. So once those intron sequences are in place and you've got your messenger RNA, you then have a series of post-transcriptional modifications where you get your poly A tail, you've got your mature RNA after a series of methylations. You then get, and you get removal of the introns, you then get your mature messenger RNA with your exons of interest, and then you will get translation of that mature RNA, typically by those ribosomes sitting at the endoplasmic reticulum. This protein or polypeptide then gets put into the ER, and during those process from the ER to the Golgi to the endocytic system to the plasma membrane, you've got a series of the post-translational modifications that I mentioned to you, some of them being peptidases, as some of these are produced as prohormones. This then leads to usually cleavage of a signal sequence, signal peptide. It then gives you an active hormone, and then finally, typically, some sort of peptide fragment. That this peptide fragment we'll talk about shortly, but in some cases, especially in the case of insulin, can be used as a marker for insulin levels or insulin production. Because oftentimes these peptide fragments are secreted, so you can use and measure the peptide fragment to know how much of your active hormone was made in your body at that time. So this is the general flow for peptide hormone synthesis. And obviously, if I ask you how insulin is made, I would like to see this recapitulated. Okay? But that would be true for most all of the protein hormones that we will talk about. Okay, so this is what insulin synthesis applied. So if we take those fundamental principles I just talked about and apply those to insulin, this is exactly what we get. So insulin is first made and transcribed. You've got beta chain and alpha chain. So on the same gene, you've got an intron that separates them. You've got the C chain of insulin, so it's made up of A, B, and C. And the C chain transcription side is bridged. It's two exons bridged by an intron. So you need to remove this intron and rearrange so that you actually get your C chain produced. Importantly, you have a signal sequence put at the beginning, which then when this first signal sequence is made and translated by the ribosome, it tells the ribosome, oh, I need to go target myself to the rough endoplasmic reticulum. And so the ribosome translation will stop. It will get sent to the rough endoplasmic reticulum with this large messenger RNA stuck to it, and then it'll restart the translation once it binds to something called the translocon located within the endoplasmic reticulum, and start to remake and continue with the synthesis of this insulin polypeptide. And as you can very quickly see within the rough ER, some very important steps need to occur. So you've got production of these amino acids, and right away you see these cysteine amino acids are made, and these have to fold appropriately, right? You see how insulin folds over on itself to start to form the final fold of the protein. And these disulfides are made and formed co-translationally as the polypeptide is coming out of the ribosome. And this is really to protect the polypeptide. In addition, it's going to present and allow for presentation of key amino acids to the glycosylation machinery. So there's a fine balance or dance, if you will, between protein folding of the insulin in the ER, addition of glycosylation groups, sugar groups, and formation of disulfide bonds, right? And those processes are essential to getting insulin into the endocytic system and eventually to be bound to the insulin receptor. And if they don't form properly, then you won't get secretion and you won't get recognition of the hormone onto its receptor. Importantly, the other crucial aspect of the folding is insulin gets cleaved by those peptidases that I mentioned to you, so the key peptidases. And the folding is essential for presenting these key, you see these double arginine and lysine arginine. These peptidase sites have to be presented in a specific manner so that the enzyme can recognize them. And that is due to the folding that occurs in the rough endoplasmic reticulum. Once the cleavage events occur within the Golgi as it's moving through, then you start to get what looks like a mature insulin hormone made up of this alpha and beta chains. Now remember when I talked to you about extra polypeptide regions which can be monitored, well, insulin's a perfect example of that, where you've got this C-peptide that comes out after cleavage, and you can nicely monitor C-peptide as a reporter for production of insulin, active insulin, because they all came from the same gene transcription, gene translation polypeptide in the ER. So if you measure C-peptide, you clearly know that A and B were made simultaneously, and you can use that as a readout for how much active insulin is being produced by that particular beta cell. Okay? And then eventually, obviously, this active insulin and C-peptide are, as I mentioned to you, move out of the Golgi. They're packaged into those final vesicles in the endocytic system, and the secretory vesicles, remember those dark circles that we saw in the optimized cell type, will then move to the plasma membrane, where it will then secrete these hormones into that blood, which is then flowing from the top of the pancreas down the center out and enter the bloodstream. Okay, so it's a very finely tuned production process that is going to deliver insulin to the rest of the body. And when we start talking about things that go wrong, understanding the process will help us appreciate how sensitive the whole system is to perturbation. Okay, why don't we take a break, and we will come back in, let's say, shall we say 8:15? Or no, 8:15, that's not your time. We have to, sorry, my brain is need another coffee. How about we come back in 25 minutes? That sound good? Okay.